So we've looked at the different types of signaling molecules. We've had a look at the stimulus response model, so our reception, transduction, and cellular response. And we've looked at how different signals will have receptors placed in different areas in the cell, depending on whether the signal is hydrophilic or hydrophobic. So we're going to move on to these last 2 dot points. I know this one looks like a lot, but it's just got quite a lot of information written in here. But this is basically looking at apoptosis, which we did look at in year 11. So we're just going to look a little bit deeper at that. And then also what happens when apoptosis goes wrong. So hopefully you remember that apoptosis is the controlled death of cells, or sometimes you hear it referred to as programmed cell death. So this is a normal thing that happens in all living organisms, and it's actually a really important process. We need cells to be killed basically so that they can either be replaced with new cells, or we get rid of dangerous cells. So one of the first reasons for apoptosis is to remove damaged cells. And when we're looking at cell damage, it's usually damage to the DNA. If, for example, when a cell is dividing and the DNA doesn't copy itself properly, or there's something wrong with it, we don't want those cells to divide. So apoptosis would occur in a cell that is damaged. Apoptosis is also really important in fighting disease, and we're going to move on soon to looking at the immune system. But one way that apoptosis might occur would be if, say, a cell is infected with a virus, and the best way to get rid of that virus is actually for the cell to destroy itself. And we'll look at that again more when we look at the immune system. Another reason that apoptosis might happen is when cells aren't needed anymore. Okay, well, there's no point having cells in our body that we don't need. And a really good example of this, again, we'll look at this further, is after an immune response. So let's say someone has been sick, and during that illness, their body produces a lot of immune cells to fight the infection. Once you're better, you don't need those extra immune cells anymore. So they might be sent a signal to say, thanks for your help, but you're not needed anymore, and they will be destroyed. So you should have be writing this in your notes and just have a few of these reasons of why apoptosis occurs. It has come up in the exam before where it's asked, give an example of when a healthy cell might undergo apoptosis. So that could be something like this. It's not needed anymore. For apoptosis to occur, a death signaling molecule has to be received. Now remember, this is part of the topic on cellular signals and signaling molecules. So we will look a little bit more at these molecules. So a death signaling molecule will start the process of apoptosis. Okay, so that will be the reception of that molecule will start apoptosis. And as always, it needs to bind to a receptor. It needs to have a complementary receptor. And a new word probably for most of you here that you will need to write down. So in apoptosis, we have a special type of enzyme called caspases. Remember, anything that ends in ase is an enzyme. So caspases are the enzymes that will break down all of the cell contents during apoptosis. So if you just have a quick look at this diagram up here, we've got the normal functioning healthy cell, and then it will start to break down. So eventually we need to break down all the things that hold the cell together. The organelles get broken down, and then it ends up just being these blobs basically that get eaten up, which we're going to look at later. But make sure you have caspases written down as the enzymes that break down the cell contents. Now, I said that for apoptosis to occur, we need a signal. So we need a death signal. And there's two different places, basically, that signal might come from. So when we're saying initiation pathways, we're saying where the signal, the signaling molecule, comes from. Okay, so we have two pathways. One is called the mitochondrial pathway, and that is also known as the intrinsic pathway. You should have both written down because both of these terms come up. The other is called the death receptor pathway or the extrinsic pathway. Remember, what we're talking about here is where the initial signal comes from. So we'll look first at the mitochondrial pathway. And you may know if something is intrinsic, it comes from inside. And that's exactly the case here. During the intrinsic pathway, the signal for the cell to destroy comes from inside that cell. So this happens when there is damage in the cell. Remember, we looked at a few different reasons that apoptosis might occur. If the cell is damaged, for example, there's something wrong with the DNA, the signal will come from inside the cell that it actually needs to destroy itself because it's damaged. When that signal is released, proteins will go straight to the mitochondria. Remember, it's the mitochondrial pathway. And the mitochondria will release a molecule called cytochrome C. You don't really need to know that name. You can write it down. The important thing is that the signal is coming from inside the cell. Due to damage, the proteins go to the mitochondria, and then we have what's called a cascade of reactions that activate our caspases. So remember, the caspases are the enzymes that are going to carry out the process. This is all just about switching on the process of apoptosis. The second pathway, so the second way that a cell might get a signal to destroy is an extrinsic message, or what's called the death receptor pathway. And this is when the signal comes from outside, and it's usually from another cell. And a really good example of this, and again, we'll look at this more when we look at the immune system, is when immune cells release cytokines. So remember, they're a type of signaling molecule. When certain cells in the immune system might release cytokines, and they will go to a cell that might be infected and tell it that it needs to destroy itself. In this instance, because the signal is coming from outside, it needs to have a receptor. So when this pathway occurs, the signal is received by a death receptor protein on the cell membrane. I've got a diagram on the next slide to explain that. So signal is received by a membrane, by a receptor in the membrane, and then similar to how we looked at those secondary messengers, when you have a hydrophilic signal that attaches to a receptor on the membrane, the death receptor protein will again begin what we call a cascade of reactions that will switch on the caspases. Okay, so in both of these, we've ended with the same thing. We've ended with caspases getting activated, and that is what we need for apoptosis to occur. So just a diagram to show these two pathways. So in the mitochondrial pathway, we might have some damage in the DNA inside the cell, and signaling molecules will get released to indicate that the cell is damaged. So they will travel to the mitochondria originally, And then the mitochondria will send a message here to our caspases. Okay, and once the caspases have been switched on, we've switched them all on, then apoptosis will begin. In our second example, there's nothing technically wrong with the cell. There might be a virus in there, but the cell might actually be otherwise healthy. There's no damage to the DNA. So in this instance, a signal comes from outside. So a T cell is a type of immune cell. And we'll look more at these in a few weeks. This is a cell, and this immune cell might know that this cell is infected, and so it will release cytokines usually, and they will bind to our death receptor. Remember, cytokines are hydrophilic, so they can't go straight into the cell. So they'll bind to that receptor, and then this receptor will send messages to the caspases. And caspases will get switched on, and apoptosis will occur. Okay, so you might want to draw a diagram like this. There's also a nice little summary diagram on page 284 at the bottom in your textbook. Just note that in the diagram in the textbook, it shows both of the pathways in one diagram, where actually you wouldn't get both pathways happening in the same cell. It would be one or the other. So once that signal is received, whether it comes from inside, so whether it's the mitochondrial pathway, or outside, the death receptor pathway, the steps of apoptosis are the same. Okay, so the signal is different, but then the steps of apoptosis are the same, and you do need to know these. I have some diagrams for this that it will show up on the slide, but you might, if you Google this, you'll find lots of different ways of drawing apoptosis, as long as you're getting the key information. So once the signal is received, the caspases are activated. And that's what we just went through on the last two slides. So we switch on these caspases, and they will start moving around the cell and digesting proteins. So they're basically breaking up the contents inside the cell. So we might have thousands of these little caspases moving around and starting to break apart the proteins. Step 2, the cell contents will be digested. Okay, so that kind of carries on from what the caspases are doing. We get digestion of all the main cell contents. So things like our DNA, our organelles, all of that will start getting broken down. Then the cell will start to shrink. And that makes sense. If all the things inside are getting broken down, the cell and the nucleus will shrink. So you see in this diagram, the cell has gotten a lot smaller because all of the things inside are kind of getting broken down into little pieces. And hopefully you remember this word from year 11, one of my favorite biology words. We get blebbing and breakage. So I'll just bring up the image here. So when the cell is breaking down, the membrane will start to fold and form these little things called blebs. So blebs are basically just those folds or those bulges in the plasma membrane. And then once they have blebbed enough, and let's say, for example, this part ends up connecting with this part, it will break off. So kind of like how vesicles break off, that will break off like these have and form apoptotic bodies. Sounds like a bit of a mouthful, but if you get your head around the word apoptosis, it's quite similar. Apoptotic bodies. So they're now small individual pieces. And once that has occurred, a message will get sent to other cells called phagocytes, Again, we're going to look at these more with immunity, but they are cells that will come along and basically engulf those apoptotic bodies. And that will be by the process of phagocytosis, which is a type of endocytosis. So we'll get here the phagocyte, which is a cell. So the phagocyte will come and basically digest these pieces until eventually all of them have been digested and the cell is gone. Okay, so you might want to pause this video here so you can write down these steps and do take a few minutes to do diagrams and make sure you can connect what you have written down with what is occurring in the picture. This is another representation of the same thing. Like I said, if you Google apoptosis, you'll see lots of different diagrams of it. But basically, we would have the caspases would get activated. So they would start all of this. The cell starts to shrink. The DNA has been broken into pieces. We get the forming of the blebs here. They break into our apoptotic bodies, and then a phagocyte will come along and consume those. The study design also emphasizes that you need to, with your understanding of apoptosis, you need to be able to explain what happens if either too much apoptosis occurs in an organism or too little. Okay, and it might be a good idea to pause for a second and just think what might happen if we've got too much apoptosis, so we're getting too many cells are being killed, or not enough apoptosis, so not enough cells are being destroyed. What kind of diseases might occur? So with too much apoptosis, basically healthy cells are getting a signal to self-destroy when they shouldn't. Okay, and if you think about that, we've got too many cells being killed off, and if those cells can't be replaced, so for example, neurons or brain cells, they can't ever be replaced. So if you get enough cells destroyed and they can't be replaced, that can lead to things like Alzheimer's disease or Parkinson's disease, which are all things that affect the brain. So in this image here, you can see on the left here, that's a healthy brain. And then this is a brain with Huntington's disease. If you look around the outside, you can see all these big gaps where the brain has actually started wasting away because too many brain cells are being destroyed when they shouldn't. On the other hand, if we don't have enough apoptosis, that is also extremely serious, because what happens is we might have cells that are damaged, and that happens quite regularly, that DNA might malfunction and a cell would then destroy itself. But if that cell doesn't get the signal to destroy, even though it should, it won't be destroyed. And what do cells love doing? They love dividing. So if we have a damaged cell that's not destroyed, and then that cell reproduces, and then those cells should be destroyed but aren't, they reproduce. Eventually, when that happens enough, that leads to a mass of unhealthy or damaged cells. And that is actually what a tumour is. So tumours in cancer are when cells that are damaged initially aren't destroyed for some reason. It's usually that their signal to destroy isn't received. So damaged cells reproduce, and we end up getting a mass of kind of unhealthy cells that we can see here, damaged cells in otherwise healthy tissue. So make sure you've got some notes down on this, so you could explain how too much apoptosis can be a bad thing, but also how not enough apoptosis can be really bad for an organism. Okay, so that is both of those dot points there. You'll probably notice as you get to the end of this that all of what we've covered in cellular signals really ties together quite a lot. So hopefully if there were some things early on that maybe were a little bit confusing, hopefully having gone through these other topics has helped to clarify some of that. If not, please pop the feedback into the Google form. As I said, this lesson is probably not quite a double's worth, whereas Monday's work was probably a bit more. So it should even out, but that should give us a bit of opportunity to go through some things on Friday if there were certain bits that were a bit confusing or you have questions. So I need you to put information in that Google form so I can prepare maybe a few things to run through when we have our Google Meet on Friday. Otherwise, jump into Edrolo and answer the questions like I've instructed in the Google Classroom. Okay, chat to you soon. Bye.